Hello, everyone. I'd like to start by thanking the organizers for the invitation to be here today. It's really a pleasure to show you some of the work that we've been developing in recent years. So I'd like to start by maybe showcasing the approach that we've taken in some of the research that we've done over these last years, where we've basically taken a reductionist approach to understand how specific genes may modulate synapses, circuits, and behavior. So this work is often performed in the context of neuropsychiatric and neurodevelopmental disorders, mainly because this allows us to get insight into elements that may be particularly salient in the control of behavior and physiology, both in health and in the context of disease. While at the same time, we hope that this approach kind of allows us to peer into molecular and synaptic mechanisms. So I'm just highlighting here some of the contributions where we look at some of these genes, like, for example, SHANK3, or more recently, adapting proteins to the mGlu-R receptors and how they regulate specific synapses and circuits. So, but keeping with this idea of neurodevelopmental and neuropsychiatric disorder, even in the cases where there's this very strong heritability, we still have to consider that genetics may only explain up to 50% of what may be inducing these pathologies. And so keeping in line with the theme of the symposium today, something that has been preoccupying me recently and regarding the project that I'm showing you today has to do with the environment and trying to understand how environmental insults may also shape brain development and potentially even induce brain disorders. So if I need to convince someone about the importance of the environment, I often turn to this example of the rubella epidemics in the 1960s, where basically individuals that were born from mothers infected with this virus actually later on became quite at risk of being diagnosed with autism or schizophrenia. And so while the prevalence at the time for these pathologies was just shy of 1% for the general population, it actually shot up and rose up to 13% and 20% respectively. So we're now getting some very, very interesting insights from various labs across the world on how maternal immune activation may be a critical element in regulation of fetal brain development. And obviously, the environment and the immune status play a key role in shaping this normal physiological development. And so one of the key messages that has been transpiring is that specific immune insults may lead to discrete responses to patterns of cytokines that may be produced that then impinge on this normal brain development. So the part of the work that I want to share with you today is actually focusing on a different type of immune insult, namely allergies. And so this is one of these other cases that have been reported in the literature for a significant amount of time, that individuals that suffer from severe allergic illness are almost up to a 4-fold increase of risk of being diagnosed with attention deficit hyperactivity disorder. So this has been described across various countries. It's been reported since at least 1976, but the mechanism linking these two apparently very different disorders has remained largely unknown. So, but turning to the actual signaling or effector molecules in these pathologies, if we try to take a reductionist approach, we need some targets to actually go in and interact with. So in the case of bacterial and viral infections that may trigger a Th1 or Th17 response, like what I showed you before, these are some of the cytokines or elements that get produced. But in the case of allergies, the key element that I'm going to spend some time talking to you about today is actually interleukin-4. But there are two important concepts that we have to consider at this point that diverge from this maternal immune infection, such as in the case of rubella, which is the fact that allergies normally get triggered postnatally, after birth. And so there's two keywords that there's two keywords that we have to take from here. One is postnatal, and then the other obvious one is neuroimmune interactions. So if we're talking about neuroimmune interactions or the neuroimmune system, one of the key elements that we need to consider are microglia. So microglia are, as you well know, the macrophages of the brain. They support neuronal function, they promote synaptic pruning, but they also are involved in phagocytosis. And this can be phagocytosis in case of an injury where pathogens need to be removed, but it's also phagocytosis of neurons, removal of neurons from brain circuits. So if we consider the second element, which is a postnatal modification, then we probably should turn our attention to regions of the brain that actually seem to mature during this phase after birth. And so the cerebellum is actually one of the key elements in this story because it's a region that largely develops postnatally. And we actually also know that microglia in the cerebellum is particularly active if we consider, for example, the molecular signatures associated with these cells in this particular brain region, and some other characteristics that they're showing. Additionally, and this is also something that captured our interest, is that the cerebellum itself has been somewhat implicated in the pathology of ADHD. So we kind of have these different moving pieces kind of suggesting that there could be some involvement here, but there is still very few evidence of actual active pruning of microglia occurring in this brain region. And so that's actually where we started this project. So we wanted to first get an animal that would label most neurons in this brain region, in this structure, in the cerebellum. So if anyone has worked with the PV-CRE line, with any type of reporter, GFP, TD tomato, one of the things that you probably noticed as you were dissecting these brains is that the cerebellum kind of is shining at you. And so that also happens when we do this cross of PV-CRE with a Rosa TD tomato reporter line, where virtually the cerebellum is all labeled in red. And so there's actually data from single cell studies showing that indeed parvalbumin is at some point during early cerebellar development expressed in virtually every neuron that is in that brain region. So this allows us to get a system where all the neurons are labeled, and microglia, we can actually label it with immunohistochemistry or antibodies or other techniques. And so what we end up is with the preparation where we can go in and look for TD tomato particles inside of these macrophages. And so we can then use, for example, flow cytometry, where we can dissociate the tissue but still nicely capture microglia, and then look at the percentage of these cells that do contain neuronal material inside of them, or we can go for more classical studies with confocal microscopy and reconstruction and observe what is actually going on. So this is a central piece to the question that we still wanted to resolve. And this is actually work that is largely developed by this very talented postdoc, Joana Guedes, and Pedro Ferreira, student in the lab. And I just take the opportunity to tell you that this paper is actually coming out next week in Neuron. So with this tool, some of the first things that we started to do was actually ask, well, is microglia actually pruning, removing material from the cerebellum in the postnatal period? And the answer seems to be yes. So the percentage of these cells that have TD tomato inside of them is actually very high in around P10 during the first 2 weeks of development, and then it sharply decreases by P16. Now, one thing that I didn't tell you is that in those axes of immune system activation, bacteria and virus tend to push macrophages in one direction, whereas IL-4 and allergies tend to push them into a completely opposite state, or I shouldn't say opposite, but a orthogonally different state of activation. And so IL-4 is actually known to dampen macrophage activity. And so what we actually found when we started looking at the IL-4 levels in the cerebellar parenchyma is that these were actually increasing with time. So when we plot the two aspects together, pruning and IL-4 concentration, there's this very nicely invertly proportional curve. So it seems that physiological levels of IL-4 could be somehow related to microglia engulfment of neurons, and kind of made us ask this question if IL-4, physiological IL-4 could be dampening microglia activity. So then what we decided to do was to go in and perturb the system. So we perturbed it into basically two ways. So using LPS, which is a bacterial lipopolysaccharide, we can kind of push it towards one specific axis of immune activation, making microglia or macrophages more active, and IL-4, we hope that it's pushing it into the different direction. And so what we see, and this is data from this flow cytometry setup that I mentioned, is that in control conditions, if we do this analysis at P10, there's again very significant engulfment of neurons by microglia. But if we administer IL-4 to these animals, we actually cut this down almost by 50%. What is also very interesting is that LPS seems to have no significant effect at this age, suggesting that the level of activity by these cells is already capped out. It's already maxed out. We did the same or similar analysis using confocal microscopy, where we just go in, find microglia, and then count the ones that actually have these particles inside. And again, as we can see in the condition with IL-4, this suggests that there's a much lower pruning being done by these cells. So one aspect that we started to attack, or that we actually imagined in our minds when we began this project, is, well, maybe microglia is doing this more classical synaptic pruning. So this was an ongoing story at the time, and we were thinking that maybe microglia is going in and removing particular synapses. Another competing hypothesis would be that microglia is actually engulfing entire neurons. And what pointed our thoughts in that direction were actually the size of the particles inside. And in some cases, I probably should have put in an image here, we can actually see microglia engulfing entire cell bodies. So if this idea of engulfment of neurons and removal of neurons from the cerebellar circle actually pans out, what would be a prediction? So one prediction would be that, for example, the connectivity between cerebellar granule cells that have these T-shaped axons synapsing to Purkinje cells would be enhanced if somehow these cells are escaping their pruning. And so we decided to use patch clamp to record activity in Purkinje cells and get a sort of measure from this activity. And what we actually found, again, mimicking the previous results, is that if IL-4 is administered at P8 and the analysis done at P10, P13, P15, P28, and then P30, we only we see this massive increase in frequency of events on Purkinje cells only when the manipulation is being performed at P10. Again, suggesting that we could be somehow hitting a specific critical period of development. So to then further convince us that this could be granule cells escaping pruning, and moreover, or more importantly, that this could somehow be perpetuated into development, we did similar types of recordings where we injected the animals at P8, but then did the recordings at P60, and this was largely preserved, suggesting that indeed this escaping from the pruning it becomes a permanent feature of the circuit. We also looked at the fiber volley, which basically are stimulating mostly the axons from the granule cells, and we see consistently that the fiber volley is increased, again suggesting that if you have more parallel fiber axons, you probably have more granule cells. We also looked at the granule cell density. Here I'm just showing you a gross histology image where it's clear that these sections look somewhat different, but we have more refined data that I can share with you if it becomes interesting later on. So, but then the big question is, well, there seems to be cerebellar dysfunction. Does this translate into altered behavior? And so one of the very first tests that we conducted in these animals were basically open field tests where we're just monitoring the animal activity. We also did running wheels, and in all of these tests that require that allow us to observe motor output from these mice, we could actually see that the travel distance was always significantly increased in the IL-4 condition, but we were finding no changes, for example, in anxiety, in social behavior, and in a wide variety of other tests. So another trial that we performed was actually based off of this Stanford marshmallow experiment that, kids are given marshmallow, and then the experimenter says, okay, just wait a little, I'll come back later, and if you haven't eaten the marshmallow, we'll give you a second one. So this is sort of a proxy to assess impulsivity control. And so we do something that is somewhat similar in mice. We allow them to get a greater reward if they actually wait a certain time delay. And with 5 seconds, we don't see any difference between controls in earlier stages of the test or in later stages of the test. But as the difficulty of the trial progresses with 15 or even 30-second delay, then we see that these IL-4 animals become progressively more impulsive. So some of the other questions that we wanted to explore here were, to a certain degree, to understand if indeed IL-4 is both specific and if it's actually, if this response is truly being mediated by IL-4 interacting with microglia rather than with, for example, other cell types or even neurons, where we know that these cytokines can also alter synaptic activity. So we first went for a more naturalistic allergy model. So we employed house dust mites. We used a similar time frame. This protocol is somewhat different. It was developed by colleagues working in the immunology field, where we intranasally administer this cocktail of mites in PBS, and we have the animals inhale this for a few days. We saw very similar phenomena in terms of the cell density in the cerebellum, similar phenomena in both electrophysiology, and even more robust changes in terms of the animal behavior. So we were very happy with that, and so we decided to also test if we ablate the interleukin-4 receptor in microglia, if we could actually block this phenotype from appearing. So we developed this, we crossed these animals, the IL-4 receptor flox line with a CX3CR1 Cree driver line. We tested our protocol again, being a completely different strain that we were using, and we could still elicit the behavior both with IL-4, but again, more robustly with the AGDM trial. And indeed, in the presence of the flox allele, so meaning that the receptor is deleted in microglia only, we actually block this from appearing. So the story that I'm showing you is that we're fairly convinced at this point is that indeed this late maturation of the cerebellum is somehow interacting with low levels of IL-4 that are physiologically present, and this is basically creating a critical period that allows granule cells to be removed from the circuit. Okay? So what we can do is that we can accelerate, we can shift the window earlier by administering this IL-4, basically preventing the pruning from occurring. And then eventually, potentially, other cytokines and other elements will still come in, but the window has already been closed prematurely by us. And so this becomes a somewhat irreversible process. And so low levels are needed to delineate this critical period. By increasing them, we prevent physiological pruning, and so we actually end up with excessive granule cells that drive enhanced excitatory inputs into PCs, suggesting that indeed we may be in the presence of at least a mechanism that may explain some cases of this link between allergies and hyperactivity. So just to finish, I'll steal a couple of minutes from the Q&A session, but I'll try to be brief. I'm sure some of you in the audience may be thinking, okay, ADHD, we treat this with dopamine. So how could the cerebellum be linked to dopamine? It turns out that there's a very nice paper published just a few years ago by Cameron Cadocan in New York, showing that there are indeed projections, direct projections, monosynaptic projections from the deep cerebellar nuclei into the VTA. And so we decided to try to replicate their findings, and we were actually very successful in doing this. So what I'm showing you here is some preliminary data that we're still working on. So we hope to follow up that main story with at least some of these new circuit dissection that we're doing, where we infect the DCN with chanorodopsin. We can clearly observe these DCN fibers very close by to the cell bodies of dopaminergic neurons in the VTA. So now we have a model where we can, for example, just using patch clamp, detect this type of pacemaking activity that is somewhat typical of these cells, but then we can actually stimulate the system to induce currents and stimulate firing of action potentials. Another aspect that this allows us to do is to study characteristics that may be specific in particular synapses. So we know that this type of behavior, where we see a type of short-term plasticity changes, is often characteristic of individual circuits. So in the case of the DCN to VTA projections, what we can observe is indeed a short-term depression when the frequency of this stimulation is at least higher than 5 Hertz. So between 5 Hertz and 10 Hertz, we see this very nice expression of short-term plasticity. And so we obviously employed this strategy in the context of our IL-4 animals. And what I want to share with you today, and again, keep in mind that this is still very preliminary, is that, first of all, we see a very, very significant reduction in the invoked amplitudes, basically just shining the light and then recording from the neurons in the VTA. However, I'm always skeptical about these results because we could have problems with the infection. There could be a whole slew of confounding effects. However, this type of N1 over N2 response with the peripulse depression kind of alleviates that. So from this data, I'm actually somewhat more excited about these potential changes in plasticity. But so we still have to reconstruct the circuit. We still have quite a lot of work to do both within the cerebellum and downstream of the cerebellum to understand if indeed this is somehow, this alteration in its brain region is somehow impinging on dopaminergic activity. And so just to finish, these are some of the type of big questions that the reductionist approach then is not very good at handling. And these are the questions that actually keep me awake at night. So we have this notion of the hygiene hypothesis, right, there, where we think of allergies as a disease that will show up that you cannot prevent, an unpreventable disorder. But indeed, there's an hypothesis suggesting that in hypersterile environments, there's a stronger probability of developing some of these pathologies. And so this has preoccupied immunologists for a long time, and what is preoccupying me is, what if this actually is impinging on brain development as well, which I think is a very interesting question for us to consider. And then the second question is, thinking outside of the context of disease, could it actually be possible that this change in behavior by an allergic agent could somehow subserve an ethological role in the context of normal behavior by the animals? And so I'm constantly thinking about this, but I would love to hear your thoughts on this work. And just to finish, I have to thank the members of the team, but this is, again, work largely developed by Joana. Pedro, Ana Luisa is a senior postdoc and a very good colleague and friend in the lab, and she's instrumental in developing this work. And all of our collaborators, but mainly Zip, who was very important in helping us drive this project, and obviously the funding agencies. So thank you very much.